Hello student, myself Marudarshana from Matrushri Ramuba Tejani and Matrushri Santaben Vidya Science College, which is connected by Sardar Patel Educational Institute. Today we talk on Sem number 3, paper number 303, name Microbial Physiology and Metabolism, topic name Enzyme. Including this topic, we discuss on the definition of enzyme, structure of enzyme, activity of enzyme, location of enzyme, as well as nomenclature and classification of enzyme. So focus on the topic name enzyme. Enzyme is chemical reaction is biological system rarely occur in absence of catalyze. The catalyzed are specific protein called enzyme. A catalyzed is a substance that speeds up a chemical reaction but isn't changed by the reaction. The enzyme catalyzed all aspects of cell metabolism. Enzyme is biological catalyzed. Now we'll talk on properties of enzyme. Enzyme are protein like simple and conjugated protein. Hydrophobic and colloid in nature. Denaturation is the change in structure, loss of activity by pH and temperature. Remain unaltered in end. Requiring some small quality. For example, a single enzyme can act upon 5 lakh substrate molecule per minute. This value is known as turnover number. Effect of temperature. Enzymes are so sensitive to heat. They are destroyed by high temperature. Above 60 degrees Celsius, the enzyme coagulate and become inactivated. Reversibility of enzyme action. Action are reversibly. For example, glucose 1-phosphate converted into glucose 6-phosphate by enzymatic activity. But glucose 6-phosphate again reversible enzyme activity then form glucose 1-phosphate. Specificity of enzyme. Each enzyme will only one type of substrate or group of related substrate. This is called specificity of enzyme. Now we talk on chemical properties of enzyme. Enzymes are specific, we know. Each enzyme usually catalyzes only one reaction. Enzyme substrate complex. Before form the product, the enzyme will bind with substrate and form an enzyme substrate complex. For this, and after this reaction, the enzyme will remove and product are formed. So, enzymes are not altered or used by the reaction they catalyze. So, can we use again and again? Enzymes are sensitive to temperature and pH. Many enzymes need cofactor in order to function as activation group. Enzyme function may be slowed down or stopped by inhibitors. Now we look up to enzyme structure. You all know enzyme is a proteinic nature. Enzyme have mainly two sides. First one is catalytic or active site, or second one is allosteric site. The active site for attachment of substrate known as catalytic site, and this site form enzyme substrate complex. Other site name allosteric site. Allosteric site name suggests allo means other, steric means site. Binding the site other than active site is known as allosteric site. Prosthetic group. A group is bound to the enzyme protein, which known as prosthetic group. Apoenzyme. Protein portion consisting of amino acid chain. Holoenzyme. Both cofactor and apoenzyme help to form a conjugated enzyme called haloenzyme. Cofactor. activate enzyme and are not protein part, which known as cofactor. Coenzyme, a small heat stable diacylable organic molecule which readily dissociated of an enzyme protein, for example NAD or NADPH. Now we discuss on location of enzyme. Some enzymes are located in the organism. Mainly two types of location are occur in enzyme activity. Either it is extracellular or intracellular. All enzymes are initially produced in cell. Some can pass through cell layer under certain conditions. Based on the site of action, enzymes are of two types. As I tell you further, extracellular and intracellular. Enzymes which carry out metabolic reaction within the cell are called endoenzyme or intracellular enzyme. Enzymes which are liberated by the cell and catalyze reaction in vicinity of cell are called exoenzyme or extracellular enzyme. Enzymes that make necessary change to allow them to enter the cell. Now we look up to nomenclature of enzyme. The naming of enzyme is called nomenclature of enzyme. Enzymes are named based on substrate, their reaction, synthesis, chemical nature, etc. There are some group of nomenclature of enzyme. So, first group is nomenclature based on substrate. Substrate is the substance on which an enzyme acts. Many enzymes are named by adding the suffix as to name of substrate. For example, the enzyme acting on carbohydrate are named carbohydrase. The enzyme acting on protein are named proteinase. The enzyme acting on lipid are named lipase. The enzyme acting on nucleic acid are named nuclease. The enzyme acting on maltose, which name maltase. The enzyme acting on lactose, which name lactose. The enzyme acting on sucrose is sucrase. The enzyme acting on urea is urase. Second nomenclature based on reaction. The enzymes are highly specific as to reaction they catalyze, hence this had necessitate their naming by adding the suffix "ase" in the name of reaction. For example, the enzyme catalyzing hydrolases is named hydrolase. The enzyme catalyzes oxidation is named as oxidase. The enzyme catalyzing reduction is named reductase. The enzyme are catalyzing dehydrogenation is called dehydrogenase. The enzyme catalyzing phosphorylation is called phosphorylase. The enzyme catalyzing transmination is called transminase. The enzyme catalyzed isomerization is called isomerase. Third type of nomenclature based on substrate and their reaction. Some enzymes are named based on the substrate utilized and type of reaction catalyzed. For example, the enzyme removing CO2 from pyruvic acid is named as pyruvic dehydrogenase. The enzyme removing hydrogen from isocitric acid is named isocitric dehydrogenase. Fourth type of nomenclature based on synthesis. Enzymes are also named by adding the suffix "ase" the substrate to be synthesized. Thus, enzyme synthesizing citric acid named citric synthetase. Based on discover, fifth type of nomenclature. Certain enzymes are named by discoverer of enzyme. For example, pepsin, trypsin, etc. 6 types of nomenclature based on enzyme commission. Commission of enzyme name the enzyme into 6 groups based on chemical reaction catalyze. There are oxidoreductase, transferase, hydrolase, lyase, isomerase and ligase. 7 types of nomenclature based on EC number. The commission of enzyme name the enzyme by code number called enzyme commission number. In short form, EC number. According each enzyme name by 4-digit number. For example, 5213. The first 5 represent the 5th class of enzyme, which known isomerase, given by the commission of enzyme. The second number represents subclass, the third number represents subclass, and the final number represents enzyme. So this is all about the nomenclature of enzyme. Now we discuss the classification enzyme or IUP classification enzyme. Commission of enzyme name the enzyme into 6 groups based on chemical reaction catalyze. This all 6 groups we look further in nomenclature enzyme. Each class we talk on detail. First class, oxidoreductase. This class of enzyme is concerned with the oxidation reduction reaction, which is one compound is oxidized and another reduction. Oxidation involves removal of hydrogen atom or an electron from a donor to common acceptor. Such enzymes are known as dehydrogenase and oxidase of the class involve molecular oxygen as one of the reactant. For example, oxidases and hydrogenase. Second, transferase. These are enzymes which catalyze the transfer of chemical group such as alkali, methyl, carboxyl, amino acyl, sulfate, aldehyde, ketone, glycosyl, phosphate group, etc. from a donor to acceptor. For example, transferase. Third group, hydrolase. These group include hydrolytic enzyme that split CO, CN or carbon, and other bond are addition of water. This including esterase, lipase, glycosidase, peptidase or phosphatase, etc. Lyase, fourth group. These are group of enzymes which catalyze the removal of specific group from substrate and introduce double bond. Some enzyme can add group to the double bond. Important examples are aldolase, decarboxylase, dehydrolase, etc. Fifth group of isomerase. Isomerase are a group of enzymes which catalyze redistribution of channel group or chemical group, which are molecule and produce isomerase epimers, etc. Important examples are isomerase, epimerase, intramolecular transferase. For example, sixth and last group of this classification is ligases. These are also called synthetases. The catalyze the joining together of two molecules coupled with breakdown of phosphate bond in ATP or any nucleoside transfer phosphate. Example including synthetase or carboxylase. So, this is all about IUP classification of enzyme. Thank you for watching.